Welcome to our methods video on how to do a disc diffusion assay. In this video, we will explain the theory behind the assay, the basic procedure, as well as how to minimize error and potential pitfalls to look out for. Let's begin by introducing the assay itself. Diffusion assay is used as a way to quantify the sensitivity of the bacterial strains against a certain antibiotic. This assay uses commercially available long and paper discs that contain antibiotic that slowly diffuses over time. In our case, we use macrolide discs that contain 15 micrograms of each antibiotic. How susceptible the bacteria are to the antibiotic is related to the size of the zone of inhibition. The bigger the zone, the more sensitive the organism. Here is a typical setup of a disc diffusion plate. For our experiment, we used 3 controls against 3 antibiotic discs, evenly spaced out across the plate. Bacteria that are more resistant to the antibiotic will have smaller zones of clearing. In some cases, there will be no clearing at all. It is important to note that the antibiotic does not only diffuse horizontally across the plate, but vertically as well. Because of this, plate thickness does have an effect on the resulting diffusion pattern. Moving on to the basic procedure of this assay. First, begin by preparing an appropriate volume of overnight culture. We use 5 milliliters of overnight culture in our assay. Next, measure the OD660 of the culture and dilute this overnight culture to a predetermined OD, to ensure that each batch will contain a similar number of cells when the culture is plated. Be aware that higher OD readings can have increased uncertainty due to the exponential increase of scattered light. Pipette an appropriate amount of diluted culture, ensuring that it contains enough bacteria to produce a confluent lawn, but not so much that spread plating becomes impractical. For example, we use 100 microliters. Spread the culture on a plate until it is evenly distributed or the plate is dry. If the plate becomes too dry, continued spreading may damage the agar. The use of a Bunsen burner creates a convection air cell that prevents contamination due to background aerosols. Use flame-sterilized forceps to place the antibiotic and control disc on the plate, ensuring that they are evenly spaced apart. You may wish to place the plate onto a pre-drawn template so that you know where to place each disc. Invert and incubate the plates at an appropriate temperature for the specific bacteria strains used. For the E. coli strain that we used, it was 37 degrees Celsius. After appropriate incubation time, measure the zones of inhibition from the edge of the disc to the edge of the zone. Minimizing error. One of the major sources of error is the slight variations that can be due to the thickness of the agar, particularly in hand-pour plate. For example, thinner plates may have larger zones of inhibition due to the antibiotic diffusing faster horizontally in the reduced agar volume. Conversely, thicker plates may have smaller zones of inhibition due to antibiotic diffusing further vertically through more agar, reducing horizontal spread of the antibiotic. To ensure that all the plates are the same thickness, we recommend using machine pour plates. Common pitfalls in this procedure are the addition of excessive amounts of bacteria from insufficient dilution schemes or larger volumes of culture added to the plates. Excessive bacteria can influence other variables, such as biofilm accumulation, which may affect your results. Using higher volumes of a sample makes the plate harder to work with because the liquid would cause the antibiotics to not adhere properly, and additional time is required for the liquid to evaporate, potentially increasing the risk of contamination. Additionally, the increased volume of liquid will dilute your antibiotic concentration, causing the data to not be accurate. Another consideration to be aware for penicillin-derived antibiotics are satellite colonies that can grow around an antibiotic-resistant colony. The resistant colony secretes beta-lactamases that degrades the antibiotic and allow some susceptible cells to survive the assay. Thank you for watching our video presentation, and best of luck in your experiments.